Hi, my name is Aviya and today I'll be sharing some of my PhD research at Deakin University. I'd like to begin by acknowledging the Wurundjeri, Bunurong and Gunditjmara as traditional custodians of the land on which this research took place and pay my respects to elders past, present and emerging. When we think of conservation, we often approach this from a single species perspective, such as conserving a threatened species. We might also consider this from a multi-species perspective, such as our efforts to reinstate past mammal assemblages. But a species isn't just an island, and they exist alongside other species and within the environment itself. Most importantly, there are these many and varied interactions that exist both between species and with their environment. And it's these interactions that form an interconnected web that really binds the system together. Thus, when we lose species from our landscapes, we may not only be losing the species itself, but we can weaken or lose these interactions, which can further contribute to declining ecosystem health. Conversely, if we can reinstate these species, we may be able to reinstate these lost interactions, and in doing so contribute to restoration. One such interaction is that between mammals, plants, and mycorrhizal fungi. Fungus-feeding mammals use fungi as a valuable food resource. By feeding on fungi, they help disperse fungal spores, both during foraging and further afield through their scats, which is an important mechanism of long-distance spore dispersal. Now, this animal-mediated dispersal is particularly important for truffle-like fungi that fruit underground and are otherwise unable to actively disperse their spores. Now, this dispersal helps with genetic mixing and the establishment of new fungal colonies. Many of these truffle-like fungi are mycorrhizal, which means that they form symbiotic associations with native trees and shrubs, and this can help plants access water and nutrients, as well as improving pathogen resistance and drought tolerance. In turn, the plants provide the fungus with carbohydrates and provide habitat and additional food resources for the mammals, completing this three-way interaction. In addition to dispersing fungi, the action of digging itself plays an important role in our ecosystems and has led these mammals to be classified as ecosystem engineers, species that impact the availability of resources, both themselves and other species. Now, through this digging activity, they can also influence soil properties, help capture seeds, provide germination niches for plants, and create habitat for other species, such as invertebrates or reptiles. And it's these interactions combined that really contribute to ecosystem function and health. But if we know that ecosystem function is this key puzzle piece when we're trying to restore our landscapes, the next question really becomes what mechanisms, approaches, or tools do we have to support or reinstate ecosystem function within our landscapes, and what role might different species play within this? With that question in mind, my PhD research aims to understand how digging fungus-feeding mammals contribute to ecosystem function, and in doing so may act as catalysts, supports, or accelerants of restoration. And today I'll be sharing my research set within a translocation and revegetation context. Firstly, I've been using eastern-barred bandicoots as a case study to explore how translocations may have dual benefits for both species conservation and the reinstatement of ecosystem functions. I did this by comparing fungal composition within eastern-barred bandicoot scats across translocation sites. Eastern-barred bandicoots are known as fungal generalists, meaning that they use fungi to supplement their diet, largely when fungi are most abundant in the landscape, such as in autumn and winter. Now, my study focused on 5 translocation sites in Victoria, 2 inside the species historic range within fenced reserves, which are shown in blue on the map, and 3 on fox-free offshore islands outside the species historic range, shown in green on the map. Now, we collected scats from the base of cage traps and used DNA metabarcoding to target the identification of ectomycorrhizal fungi, which are those that form symbiotic associations with native trees and shrubs. Now, we identified 103 ectomycorrhizal taxa, spanning 26 genera. Of these, there were 9 genera which are known to be exclusively truffle-like and fruit underground, suggesting that bandicoots may be performing an important mechanistic role at some of these sites. We also found that there were differences in the fungal communities between sites, and interestingly, bandicoot scats from French Island, located outside the species historic range, was found to be the most diverse. This may, for example, be due to influences of fungal availability, historic land use at different sites, or the vegetation community. and we're continuing to explore these elements through further analyses and research as we speak. Secondly, I've been exploring the relationships between mammals, fungi, and plants through a glasshouse experiment. I used 2 of these eastern-barred bandicoot translocation sites and compared the bandicoot with a remnant species at each site, the swamp wallaby on Phillip Island, which is also a fungal generalist, and the long-nosed potoroon on French Island, which is a fungal specialist, meaning that they rely on fungi for the bulk of their diet. Now, I used scats to form an inoculum slurry and then added this to seedlings to see if it successfully introduced beneficial fungi and improved plant growth. I used the most common tree species at each of the sites and had 5 treatments per island, which included a fresh scat of each species, a nutrient control where the fungi had been sterilised but the nutrients remained, and an experimental control where we didn't add anything to the pots. Now, the 7-month-long glasshouse experiment has now concluded, and my next steps include assessing roots under a microscope to determine how fungal colonisation has been influenced by different treatments, and assess the fungal community that's within the roots of seedlings via DNA metabarcoding. Overall, my research is really helping to move us along this trajectory of conservation approaches as we move towards an ecosystem-wide conservation and restoration lens, where we can begin to view both the conservation and management of species from a functional perspective, which incorporates and manages for these key species interactions and ecological functions. By better understanding what role digging, fungus-feeding mammals play within our landscapes, we can better understand how conserving and reinstating these species can contribute to and also drive restoration, and the flow-on effects that this can have for both ecosystem health and rebuilding ecosystem resilience. Thank you for listening and for supporting my research project.